podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. You are about to listen to the full episode, but there will be 1 to 2 short ads immediately following this before the conversation. If you would like to hear ad-free episodes and get early access before they release more widely, become a paid subscriber at the Mind and Matter Substack at mindandmatter.substack.com. Becoming a paid subscriber is a way to directly support my efforts, allowing me to make more episodes and higher quality episodes, and also helping to free up my time so that I can spend more of it on doing research and synthesizing results from the podcast and from the literature so that I can do more things like writing on the Substack, putting more content into the newsletter, and getting these things out more frequently. So if you want to support the show and get ad-free episodes, go to mindandmatter.substack.com. Hey everyone, a great way to support my efforts is to visit my support page at mindandmatter.substack.com. A link to that page is in the episode description, or you can search for it on the Substack page itself. The support page has an up-to-date list of my affiliate partners. These are companies I work with, and if you buy those products through those links or using the discount codes provided, those will get you a great deal on a variety of products related to optimizing physical, metabolic, or mental health and vitality. These are all products I use myself, and some of them are directly related to podcast episodes or formulated by Mind and Matter guests. For example, there's a great product called Keto Citra, formulated by a kidney biologist who is on episode number 186. It contains the ketone body BHB together with several minerals in a precise ratio formulated with kidney health in mind. There are also links to products like physical technology devices to track your metabolic health, digital applications to help you find and learn about food and consumer products, and more. One product I use every day is the AquaTrue water filter. I recommend this to everyone because it gets everything out of your drinking water: heavy metals, microbes, endocrine disruptors, microplastics, you name it. Other charcoal or gravity-based filters that a lot of people use don't get everything out, they just get some of the stuff out. So I really like the AquaTrue product. You can look for a link to my affiliate partners on my support page or directly in the episode description to this episode, or you can go to mindandmatter.substack.com to find it there. All right, Professor Kristin Stanford, thank you for joining me.
Thanks.
Do you want to just start off by telling everyone a little bit about who you are and what your lab studies?
Yeah, that's great. So I'm Kristin Stanford. I'm a professor in the Department of Surgery at The Ohio State University. And our lab really works on how exercise can improve metabolic and cardiovascular health. And so we look at across a couple different models and look at different paradigms to look at exercise as really a mediator of whole body health.
Yeah, and I think everyone intuitively knows, right, that exercise is good. You want to exercise some amount. There's many different ways you can exercise. But I think the more interesting question for us here and for you in your research is, well, exactly what are the effects of exercise on health? What's going on under the hood? And what are the health benefits of exercise at the level of cells, tissues, and what's actually happening there? And you study some really interesting aspects of this. You study how exercise in parents can affect the health of their offspring, their future health. So this gets into topics related to epigenetics and intergenerational inheritance. But let's just start at a high level. In general, right, exercise is good for you. Everybody knows that. Of course, there is such a thing as too much exercise, and there's all different forms of exercise. But when we think about the question of how exercise in one generation affects the health of the next generation, before we dig into the mechanics of how that works from all the animal research that people like you do, let's think high level here about like what do we see in the epidemiology literature and what do we know in humans at a high level? What sorts of associations are out there between how much your mom and dad exercised and what they did and how that seems to affect future generations? Is there sort of a lay of the land you can give us there in terms of the general associations?
Yeah, so I mean, you're right. Like right off the bat, exercise is good, and so we started to kind of looking at some of these questions. That was kind of our overall takeaway was exercise is good, but how is it good? And how could people who maybe could not exercise at certain points in their lives, how could we still confer the benefits of exercise to those people? From the human perspective, from an epidemiological perspective, there's a good chunk of data out there that shows that parental exercise is good, and there have been studies done looking at both moms and at dads, and how exercise can affect their offspring. One of the things that's a little bit tricky in human studies is that typically if a parent exercises or a parent is very active, their child's going to be more active. So it's kind of hard to parse apart the effects of the parent versus the child. In an animal model, we can control all those things. And so that, allowed us to kind of parse out some of the questions of looking at, directly the effect of the parent exercising either prior to conception or during gestation, and how that directly could affect their offspring.
And so do we know, like, I mean, obviously the obvious guess would be, if your parents exercised regularly versus if they were completely sedentary and didn't, that's probably going to affect your health, or that's probably going to affect your children's health. Is there any data to suggest that? Anything like, I don't know, twin studies where they're separated at birth or anything that sort of point us in a direction of what exactly the difference might be for the next generation?
Yeah, that's a great question. I'm not aware of any twin studies that have looked at things like that. When we started working on this field, this was probably about 15 years ago, and I was working with Lori Goodyear. I was a postdoc in her lab, and when we started to pull some of the human literature, a lot of the studies were retrospective. They were correlative. Like, did you exercise during pregnancy? And exercise was broadly categorized as really any type of activity.
Yeah.
And since then, there have been some much more in-depth studies. A group at the University of Copenhagen, they ran the FitMom study. A group at the University of Arkansas has done a couple of exciting pieces there. And a group at ECU has done a couple of studies there. I'm sure I'm missing some, but I know those three groups have really been focused on this. And they've controlled for exercise in the moms during pregnancy, and then have in most cases started to track the children. I think the groups of children are maybe between 2 and 5 years old right now. And so I think we'll learn a lot from some of those controlled studies. I think on average, there are correlations. And so correlations that show maternal obesity is correlated to childhood obesity, paternal obesity is correlated to childhood obesity. And then more mechanistically at an animal level, we've done some modulations with maternal or paternal diet and exercise, and can really kind of show how those changes are affecting the offspring. And again, in an animal study, we can take those offspring out for about a year, which correlates to about around 45 to 55 years of age in a human. So really a good chunk of their lifespan.
Yeah. So of course we have these associations in humans. If you have obese parents, you're more likely to be obese, and that's not too surprising. And then of course, we can't really infer cause and effect from that in humans, right? Is it because the children inherited that obesity to some extent, or is it just that they're in an obesogenic environment, and so they're going to behave the same way that their parents did and eat the same types of foods? So what you guys do with animals is you can sort of parse out cause and effect and look under the hood and really sort of control for these things in a way that's really hard to do in humans. So let's talk a little bit about your research at a high level. How do you study this stuff at a high level? What's the model, and what's the basic setup for looking at something like exercise in rodents?
Yeah. So, when we first started to look at this specifically with parental exercise, what we knew and I think found like that was both expected and surprising was that exercise is so multifaceted. It affects so many things. And so the first way we set the study up was we were looking at maternal exercise, and we had I think we had 8 groups of mice to start out with. So we had moms that were chow fed or high fat fed, and we used a pretty moderate high-fat diet. So in this case, a high-fat diet is 60% kilocalories from fat. We only put them on that diet or gave them access to exercise wheels 2 weeks prior to pregnancy. And our rationale for that, so gestation in a mouse, they're only pregnant for 3 weeks. Our rationale for that was just to do a moderate intervention, right? So we weren't having them on a high-fat diet their whole lives. We weren't having them exercising their whole lives. We wanted to see if we could essentially intervene in a period prior to conception. So we had a group of mice that were exercised both 2 weeks prior to conception and throughout gestation. A group of mice that exercised only before conception, a group of mice that exercised only during gestation, and then a group of mice that exercised or that did not exercise at all. And then those
Yeah.
were either a chow or a high fat diet. So we had 8 groups to start out with.
Yeah. And how do you get the mice to exercise? Presumably there's not a little bench press in the cage. What do the mice do for exercise?
I would like a bench press. I think that'd be a nice touch. We usually use a wheel cage, and so in this case, these were all wheel cage, and mice love to run in a wheel cage. There have been some other studies that had shown, like, if you gave a mouse access to like a maze or a wheel cage, they pick the wheel cage, and so we saw that the mice ran really well. They ran an average of about 8 kilometers a day. And so we were able to really track that. You can also use swimming as a model. You can do some resistance exercise in a mouse model. You can have them kind of climb a ladder with tail or with weights attached to their tail. But by far the easiest is the wheel cage, especially because mice are nocturnal, so they like to run kind of overnight. So they're do that at their leisure.
Yeah. And so it's not only easy to do, but they do love it. And I mean, this has been dissected by neuroscientists. Like, they actually enjoy it. So it's sort of a natural behavior, natural exercise behavior. They're motivated to do it. It's rewarding. It's a good assay in that sense as well.
Yeah, absolutely. And we, in a subset of these mice, we did some other stuff with a mouse that were missing a specific gene, and those mice did not like to run in a wheel cage. And so we actually repeated all this stuff with treadmill exercise. And the phenotype in the offspring is very similar. And it just went to show that those mice would run. That was a special subset that did not like to, but that's very, as you said, it's very rare. Most mice really just love to run. So it makes it an easy model for us to be able to use.
Okay, great. And so before we sort of dig into some of the experiments you've done, the idea here is, the parental generation can do exercise, say, or not. And that will have an effect on subsequent generations. So we're going to get into things like epigenetic inheritance. And I just want to give people some concepts for how this can work in principle. So when we think about a mom or a dad doing something, and then that's inherited in an epigenetic fashion to affect the offspring biologically, what are some of the major ways that these types of things can happen in terms of how cells are changing? So when we talk about things like DNA methylation, histones, gene expression, that stuff, what are some of the major sort of ways that behavior in one generation can lead to cellular and gene expression alterations that end up affecting the next generation?
Yeah, it's a great question. And I mean, I think, we think that we know, but I think there's a lot we don't know about some of these things. And so, what we've looked at in response to exercise from the dads is we've looked at their sperm and we've really been focused on the small RNAs in the sperm. A lot of other groups have looked at DNA methylation patterns and different histone modifications, but we found some really exciting things looking at the different small RNA patterns in sperm regulated by both diet or exercise. And in the moms, there's the idea is that you could make changes in the oocyte, which would then be passed down to the offspring. And one of the other things that we're seeing in the moms, which I think is kind of an interesting idea, is, is it really an epigenetic change, or do you have a healthier mom who's having, the offspring develop in a healthier environment, and is that conferring its own changes? And I think that's a really interesting question that we've not, kind of gotten to the bottom of yet. There had been some earlier studies, not our work, I believe it was Kelly Moley's work, who had shown that exercise did not have a direct effect on the oocyte, but diet did. This was, again, in an animal model. So we're not quite sure how exercise is kind of inducing some of those modifications to be passed down from the mom. Some of Lori Goodger's work showed that there were changes in the placenta or factors released from the placenta in response to exercise that could affect development of the offspring in utero.
Yeah. And so when we think about epigenetic inheritance in mammals, there's sort of a few different layers, I think, about this. And then you have to kind of do it for males and females in parallel, both on the parent side and the offspring side, for reasons that we'll come to. But when we think about paternal exercise and the effects of the dad, you're basically going to talk about the sperm there. And so we'll get to that, like maybe exercise is changing the sperm, the genes they're expressing, their mitochondrial health, that type of stuff. With the mother, you've got a couple more layers, right? You've got the actual sex cell, like maybe the egg itself is changed by mom's exercise patterns, and then the offspring literally inherit that because the egg, the oocyte itself was changed. But then you've got, right, the in utero environment throughout all of gestation. Maybe exercise before or during gestation can affect that environment. And then, of course, after birth, you've got things like mother's milk and how that is affected, and maybe the nutrient composition is different. Let's start with dad here. Let's start with the paternal side here. When you guys do studies, with mice and you're looking at paternal exercise, you mentioned you look at how the sperm change. You mentioned non-coding RNAs and small RNAs. Let's dig into some of that. But can you explain first, what are small non-coding RNAs for people that don't know?
So they're messengers. So they're talking to the different cells. They're talking they're going to talk to the offspring, and they're going to affect numerous pathways in the offspring cell, and then kind of throughout development. I think we still don't know. I mean, we know a lot, but we don't know exactly how everything is working in some of those contexts. But we know that they're given a message, and so they're changing something. And one of the things that we have seen in this. This is one of the important things I think with exercise is we see a lot of these things change. And what we don't know is if it's one that's causing any effect, or if it's 6 of them have to change in parallel to cause an effect, or if 3 of them do it, or how really that's working, or if one causes a change in one thing and 2 cause a change in some other phenotype that we're seeing. So when we started doing this, we first kind of took the broad approach to that, to look with the dads, we had groups of chow-fed or high-fat fed mice that were either sedentary or exercise. And again, a pretty moderate exercise intervention. We gave them 3 weeks of access to a wheel cage.
Got it. So basically you got mice that have the option of running. They're going to choose to run some amount. It's actually, they like to run, so they run quite a bit, but it's only for 3 weeks. So we're looking in the males here. And basically the question is, how is that exercise going to affect their sperm? How is a dad that exercises, a mouse dad, how is that sperm going to look different in certain ways compared to the non-exercising dads? And what do you guys generally see?
We see a really striking effect. So even with that 3 weeks of exercise or 3 weeks of high-fat diet, we saw a really striking effect on the small RNAs to be changed, and they were changed in different patterns. And so we started some of this work with Ollie Rando's group, and we've since I've been at Ohio State, we've started working with Chi Chen, who's at the University of Utah, and he's really interested in kind of pulling out patterns of how these sperm change. And so he's able to identify some effects of the paternal diet and of exercise, and to see what diet does and how exercise can restore it. This has been really interesting for us because there was a study that came out probably right when we were starting to work on the paternal side of things, and not to jump the gun, but what we saw in our offspring was that paternal exercise improved metabolic health of the offspring. And there was a paper that had just come out from a group at East Carolina, which saw that paternal exercise had a detrimental effect on the offspring. And when we looked a little bit closer, what they had seen is that it was again in a mouse model, but they had really intensely exercised the dads, and so it was almost an overtraining model of what they were using. And when we compared some of the small RNA changes that we saw in sperm to what they saw, they were very different. And so it was like, okay, and we were able to kind of interpret our data in the context of their data. We were seeing different changes, but with a completely opposite effect. And we've not done a whole lot. We've not really messed with our exercise paradigm to see if we could, exercise the mice more intensely, if we would see a different effect. But it was kind of an interesting observation in terms of how different exercise or exercise intensities could affect the sperm profiles in that way.
Yeah. And so let's dig into some of the details here. So how exactly do the sperm look different? These small non-coding RNAs are changing. So for those that don't know, right, the traditional RNA you typically learn about is mRNA. So a gene gets turned on, it gets transcribed in mRNA, which will then go on to make a protein. But there are many other types of RNAs encoded in the genome, and they do all sorts of different things. They can affect the expression of their genes, and they can just alter information pathways in the cell, rather than going on to be the template for a protein. So there's lots of different things that can change in lots of different ways. What are some of the particulars that you guys have observed in terms of how exercise affects these RNAs in male mice?
So we've seen a lot of the tRNAs that are really changed in response to exercise and diet. And we've looked at some previous published studies to look and see how there have been some observations of some of the small RNAs, and specifically the tRNAs that are changed. And we see the exercise kind of makes them go in an opposite manner, which is great. We always love when we can see other data that ours makes sense in the context of. And I think those have been the biggest ones that we have seen. But we've without having dug into the specific small RNAs that are changed, we were a little bit thrown by the sheer number. We were seeing like 80% of them were changed in response to just 3 weeks of exercise or 3 weeks of high-fat diet. And that was a really kind of a striking observation that's, I wouldn't consider that a hugely, it was all voluntary exercise, it was moderate at a moderate pace, but we were still seeing these really strong effects.
Right. So in other words, there's a bunch of RNAs that you can find in cells. You might see an effect in a couple of them. You might see an effect in many of them. You saw an effect in many of them. And this is what we would call a fairly moderate intervention, right? The mice can choose to run as much or as little as they want. You can give them a high-fat diet versus a controlled diet. And I guess the point is, when you guys look, you see big effects. So you give them a diet from a lifestyle change. It could be a dietary change, or it could be voluntary exercise. You're not forcing them to run a marathon or anything. And these types of interventions, dietary change, exercise, that you see big effects. You see lots of changes across lots of these RNAs.
Yeah, absolutely. And it was, I mean, we were delighted to see it, but I'm not sure that we thought we were going to see some of those changes kind of right off the bat.
Yeah, it wasn't like you saw, a couple key genes to get turned up or something like that. It's the sort of wholesale change. You mentioned the tRNAs as one that's strongly affected. What was the direction of the effect here, and what are tRNAs for people who don't know?
Yeah, so these are these transfer RNA fragments, and they were up and down depending on the diet or the exercise, and kind of depending on the small RNA that it was. And so, we saw really different effects, and some of the newer data that we have is really focused on kind of identifying patterns. And so looking at, groups of these tRNAs to change together or in opposite directions, both in response to exercise and diet. And so, I love the idea that we could pick one thing that's changed with exercise that could solve the world. But, when we know how many effects that we see in the offspring, I think really being able to look to see if these changes are they really multifaceted? Are we really seeing numerous things that kind of work together? Like 3 of them have to go up and 2 go down, and that's where we see the effect. I think so. We're still learning. We don't know exactly how these changes are going to kind of pan out. There's been some studies in looking at after a paternal high-fat diet, where they've been able to identify some of these tRNAs and inject them back into the zygote and see that has an effect on offspring development. And so that was it's kind of our initial hypothesis with exercise. We just don't know which ones we're going to have to pull out of there.
So these tRNAs are small RNAs, transfer RNAs. They're responsible basically for constructing the amino acid sequence from the mRNA. So if a gene gets turned on in a cell, it makes mRNA. It's got to go to a ribosome to turn into a protein. You've got to make polypeptide sequence from that mRNA template. The tRNAs are an important part of that process of going from DNA to mRNA to protein. So some are going up, some are going down, but this implies these changes in tRNAs you see from exercise or dietary change. This implies it's going to affect ultimately which proteins are up or down inside of a cell.
Right.
And so wide wholesale changes. You mentioned that obviously this affects offspring health. It improves metabolic health. Let's dig into that in a little bit more detail. What do you see specifically? What is specifically improved when dad exercises in these mouse models?
Yeah, so the first thing that we saw was improved was glucose metabolism. And so, to measure glucose metabolism in a mouse, we're given an injection of, we fast the mice overnight, we're giving them an injection of glucose based on their body weight. And we measure basically how fast it's cleared. And what we saw is that the offspring from the exercise trained dads, they were able to clear it much faster than the offspring from the sedentary dads. Diet played a role too. If the offspring were from a high-fat fed dad, they had impaired glucose tolerance. But even if the dad was fed a high-fat diet and exercise, it was still improved and compared to the other groups. So we were seeing, a really striking effect of exercise regardless of paternal diet on this.
Got it. And did you see that in all offspring, or is there an effect where, the daughters or the sons tend to be more affected based on what dad was doing?
Yeah, it's really interesting. So we saw a more striking effect in the female offspring from the paternal intervention. We do see it in the male offspring. It's a little more blunted. And we looked a little more closely, and we do see a striking effect on the skeletal muscle glucose uptake in the offspring, which was really interesting to us as well.
Interesting. And, I've talked to a number of people on the podcast before about this general subject, epigenetic intergenerational inheritance. And one of the things that seems to come up over and over again is you often do see sex-biased effects like this, where one sex is affected, the other one is not, or the magnitude of the difference is different. Is that generally the rule? And do you feel like we have some understanding of why that bias exists at all?
It's crazy, right? I mean, it's the first time when we started this, we were looking at the maternal effect, and we saw a stronger effect from the maternal intervention onto the male offspring, and started to dig a little bit deeper. And that's really well known, is that the maternal interventions, at least in terms of metabolism, have a more striking effect on the male offspring. and I think in animal models, I think there are a lot of things that can play a role in this. One of the things that we had seen is that we did not see any difference in litter size or in sex distribution among litters, which I thought was kind of an important caveat to make sure we were looking at some of those things. But there's some data, and I'm not quite as familiar with it, but there's some data that talks about how many male and female. So a mouse has several pups in a litter, and so how many male and female offspring that they have can make a difference, and then how those placentas sit next to each other. Right? So if you have like a male female, you might have a different response than if you have like female female. I know there's been some human studies that have looked at fraternal twins to see how they can be a little bit different. I'm not sure if anyone has looked at the interventions of the mom in those cases, but I think it's really interesting. And I think there's more for us to do and to look at in some of those cases. But I think the overall phenotype has been pretty consistent, that maternal interventions seem to affect metabolism in the male offspring, and then we're seeing that paternal exercise has a stronger effect on the female offspring.
Yeah, so it's complicated. There's lots of variables. Mice can have big litters, so it's not, it's the whole history of how many litters the mom has had and what the dad was doing and all of that stuff. But you do seem to see these systematic patterns in this realm of biology where there's these sex biased effects. So the paternal behavior might affect the daughters more than the sons and things like that. When we think about the maternal side here, let's start with the oocyte itself and then talk about in utero effects, and then talk about lactation and milk and nutrition. When you do maternal exercise, so the mom's exercise, what sort of effects does that have on the oocyte and what's going on in those cells?
Yeah, and I will say we have not directly looked at the oocytes. There have been some other studies that had looked at that, and they really saw an effect of a maternal high-fat diet on the oocyte, but less of an effect of exercise. And so that's why when we set these experiments up from the beginning, we tried to use a model where the mom exercised both prior to conception and then throughout gestation, just before conception, and then just during gestation, to see if we could parse apart really what was happening. And what we saw looking at the offspring is that it seemed to matter more. The most affected group was if the moms exercised both before and during gestation. But as long as they exercised during gestation, we were seeing an effect in the offspring. And so kind of led to us that maybe in the mom side of things, from the maternal side, we might be seeing less exercise-induced changes to the oocyte. But again, I think that still needs to be parsed out a little bit better. But that was what our data kind of indicated.
Yeah. And just thinking about the behavior here, obviously litter size matters. Like if a female mouse has a bigger litter, right, there's only so much time per infant mouse that she can allocate. There's only so much milk to go around. So litter size is going to have to matter. Does the number of litters and the mouse's sexual history matter too? Like, do older female mice tend to do like less maternal care with each subsequent litter? Are there those types of effects in terms of the sort of time investment and the attention that the mother actually gives to the offspring?
Yeah. So, and I mean, I'll tell you, for our studies, we always use virgin females because we try to. So it's just to control it the best we can, because especially with black 6 mice, they're better moms the second time around. But as you said, as they get a little bit older, they typically have smaller litters, and they're not quite as good of mothers. Kind of as you go throughout. I think they can usually breed for until they're about 24 weeks old. So we just try to control that right up front. I believe there are some studies that have looked at maternal age. I'm not quite as familiar with those, and we've not tried those interventions with exercise as well. What we typically do is we control for litter size after they're born. So we call the litters to a certain number just to account for that. So we're not, altering. So all the moms get the same number of pups, because, as you said, if you have 10 pups versus 4 pups, you're probably going to see some different effects.
Got it. So you do most of your experiments in virgin females, so they haven't had a litter before. So you're looking at the first litter they have. That provides a certain a level of control there. Makes sense. So when you do these studies, how does the in utero environment start to look different between new mothers during their first pregnancy that are exercising compared to those who are not? What does that actual in utero environment look like?
Yeah. So, I mean, when we look at the offspring afterwards, we don't see any difference in litter size or in sex distribution among litters. We're just starting now to go back and look at what's actually happening to the placenta. And I think we're going to see some exciting things there in terms of changes in vascularization and how the placenta is functioning. Some of Lori Goodger's work had shown that SOD3 is released from a placenta during exercise. And that seems to have a really strong effect on the offspring metabolic health as those offspring age. And so, I think some of those studies have been done in terms of how the actual placenta is changing and how that intrauterine environment is changing. I think, I think we're constantly learning what some of these different changes are, because everything we seem to look at in response to maternal exercise seems to be a little bit different. And I don't know that we expected that, when we first started looking at this. We're just starting with a collaborator right now with Tamar Gore to start looking at some changes in the microbiome of the mom that are happening in response to maternal exercise. And those have been really interesting as well.
Mm-hmm. So, a lot of this is TBD, but there are definitely changes. So the placenta is going to change to some extent. If we just think about this at a high level, let's just remind people, what is the placenta really doing, and what is its basic structure, to give us a sense for, like, okay, if that structure changes, what are the sort of the high level things we would expect to start to see from exercise if the placenta is changing in a certain way?
So, the placenta is where, obviously, where the offspring is developing, where they're growing. It's acting as, kind of a connection piece between the mom and the baby, and it's aging rapidly, which I thought was a really interesting kind of side note when we started talking about placentas a little bit more. So it's basically aging throughout the course of gestation, to get ready for delivery. And so, how those changes in the placenta are occurring could affect, nutrient delivery to the offspring, how they're growing and how they're developing, and then essentially as they start to be born, and then to develop from that, kind of how they respond to their specific environment.
Yeah. So obviously the placenta is the interface between the mom and the baby during gestation while the baby's growing inside of her. So naturally, if it changes somehow, in some way, it's probably going to affect the stuff that gets into the baby, the nutrients, the oxygen, all of the above, potentially to some extent. So it sounds like the details there still need to be worked out, but there probably are changes in the placenta that are happening that are induced by exercise.
Absolutely. And I think, and this might just be my lack of knowledge on it and how much exercise actually changes the placenta, but we do know that there are some, metabolomic changes in it, and there's some vascularization or vascular changes that are happening there, that have been consistent among rodents and humans throughout gestation. So I think we definitely know that there are effects of exercise on it, and we know that those can make a difference.
Yeah. When we think about after birth, so the pups are born, and now they're going to be with mom for a substantial amount of time. They're going to be getting all of their nutrition via her milk. What do we know about how exercise, before, during, and after she gives birth affects things like the composition of the milk, and how that then, has effects on the offspring?
Yeah. So, one of the things we were really interested in was how exercise could affect breast milk composition. And one of the things that we had set up in an animal model was a cross-fostering model. And so what that means is we essentially take the mice from one litter and mice from another litter, and we swap them. We have them nurse with a mom who did not give birth to them. And so when we set this up in some of our exercise trained moms, we had offspring from sedentary moms drink milk from exercise trained moms, and vice versa. And so what that allowed us to see was, if just drinking the milk from an exercise trained mom could have an effect on glucose metabolism. And the short answer was it did. And we saw really striking effects if the offspring just drank milk from the trained moms. That seemed to be enough to improve their metabolic and cardiac health as they aged. With the mouse model, it's a little tricky. So we use mice that are in really big wheel cages. And what we found was that the moms, they like to kind of bury their pups. So they would put them right in the corner underneath the wheel cages. So we realized pretty quickly we had to take the moms and the pups out of the wheel cages within 48 hours to make sure that the pups all survive. So we did not let them exercise during lactation or during their nursing period. So this was only prior to conception and then throughout gestation that they were able to exercise.
Got it. And So what are some of the effects you basically see? So the exercise-induced effects in the health of the offspring that get milk from an exercise mom, whether it was their mom or the other mom, in general, what do you see?
They had improved glucose metabolism, and they had reduced body weight. So, it was just the act of just drinking that milk was enough to improve their metabolic health as they aged. We looked at fat mass and glucose tolerance, and we looked at some of the heart health functions. So looking at ejection fraction at later time points, and really were able to see that as long as they drank the milk from the trained moms, they had improved health span.
Got it. So basically, mice that are drinking from milk from a mother that exercised, whether or not it was their actual mom or not, they tend to do better when it comes to weight and fat gain as they age, and they tend to do better with glucose homeostasis. So their blood glucose responses just look healthier.
Yeah.
Interesting. And is that like a lifelong effect?
So in a mouse, it was.
Yeah.
So in the mouse model, we did see that persisted to about, we followed the mice to 52 weeks of age, and we were able to see those effects throughout 52 weeks of age. We were able to look at a little more closely at some of the composition of the milk after that. And so, there have been some studies before that had looked at fat content and protein content and kind of how that changes with exercise. And they, not surprisingly, found that the fat content of milk was a little bit less if the moms exercised. And we kind of wanted to look a little bit differently and look at the human milk oligosaccharide content of this milk. And what we saw was really interesting to us. So in humans, there are like 130 milk oligosaccharides. In rodents, there are 2. It was done with Lars Bode, and we were looking at.
So these are just milk sugars.
Milk sugars, yeah. So they can be in humans. They can be affected by diet, by age of the moms, by numerous different factors. They are the two. The 2 that we see in rodents are 3-sialactose and 6-sialactose, and they are not found in bovine milk. They're not found in cow milk or formula. And what we were able to see is that 3-sialactose, which is like the most prevalent HMO in mice, was significantly increased with exercise. It was decreased in the presence of a high-fat diet, but if the mom exercised and was fed a high-fat diet, it still increased. And then working with Elena Andres, who's at University of Arkansas, they had data in human subjects, and they were able to look at correlations among human milk oligosaccharides and physical activity. And they saw that this 3-sialactose was increased or was correlated with activity in humans or women who were pregnant.
And do we know anything about human milk and how it changes based on the mom's lifestyle?
So we're working on some of those studies. I'm sure there's more known than I'm going to talk about right now, but working with Elaine's group, we were able to see some differences correlated to both BMI and to physical activity. And now we've got a study that will hopefully be coming out soon, working with a group in Copenhagen. This is called the FitMom study, and they had a controlled exercise intervention with these women throughout basically weeks 12 through 38 of pregnancy. And they looked at the milk, and they were able to see some changes in the milk that was kind of regulated by exercise.
Yeah. And in general, what sorts of things do they see?
So, the metabolism is a little bit different. I will say that was a really fit population of women. So I'm not sure we saw quite as many changes as we might have expected to. We have there's another study by some colleagues of ours at the Joslin Diabetes Center, and they saw in women after they had delivered, they looked at acute bouts of exercise and saw changes in specific metabolites and lipids in response to that acute bout of exercise. And one of them was this lipid 12-13-dihome, which we are selfishly very interested in because we've shown that it's upregulated by exercise in humans as well, or in different cohorts of humans. They saw that this lipid was up in milk in response to acute exercise during the postnatal stage in women.
And do we know much about this lipid and what it does or what it might be good for?
So with 12-13-dihome, we know it does a lot of good things. We know that it's correlated with triglyceride uptake in skeletal muscle and in liver. In rodents, we've seen that it has a direct effect on cardiac function, and we see that in humans with heart failure, it's down. It's increased with exercise. It's decreased in humans as they age, and then again restore with exercise. So we see that it does a lot of really good things. What it actually does in the milk, and then as it goes to the offsprings, we're not quite sure exactly how that works, but it seems to be all positive exercise benefits that we're seeing.
Yeah, and so we mentioned earlier when we talked about paternal exercise and the dad side of the equation that you often see sex-biased effects where, say, some effect will be bigger in the daughter of a father who exercised than in his sons. Do we see any sex-biased effects in the rodents on the maternal side?
Yeah, it's the opposite. So the mom's exercising or a mom's diet has a stronger effect on the male offspring.
Interesting. Okay, so paternal exercise tends to have sex-biased effect where the effect is stronger in general on his daughters, but maternal exercise, when the mom exercises, the effects tends to be stronger in her sons.
Right. And I will tell you to really complicate things, when we've looked at cardiovascular effects, the mom exercises, the more striking cardiovascular effects are in the female offspring.
Got it. So it depends on the effect you're talking about, which way the sex biased effect goes.
Yeah.
Interesting. Do we have any sense for like why that would be? Why there would be just a bias in one direction or the other?
I'm sure someone does. I do not.
Yeah.
We've just seen it very consistently and have kind of gone back and forth to make sure we weren't missing anything. Like looking at the maternal interventions, we found that when we put the moms, we basically had to put the moms on a high fat diet to see any effect in the female offspring, to kind of, stress the response a little bit.
Yeah. Yeah. I mean, I guess just off the top of my head, right, based on the way that sex chromosomes work in mammals like mice, if you're a boy mouse and your mom is the only source of X chromosome you have, maybe that's one way that some kind of bias could get introduced. The daughters have an X from mom and dad, and so those things might even out to some extent, whereas the sons are just going to get whatever genes you're talking about. If they're looking at the X chromosome, mom is probably going to have more of an effect.
Right.
Interesting.
Yeah, no, absolutely. So then what threw me with some of those were some of the cardiac effects that were then, the opposite. And so
yeah.
it's a really important point. And I think it's, I think we're still kind of learning how some of those changes take place. And I think some of the human studies, as these get further along, will be really interesting to let us kind of just give some insight into some of those effects too.
Yeah. But I guess the common denominator here is that, for any given trait you might be talking about, glucose homeostasis, aspects of cardiac health, whatever it may be, it's possible that one of your parents had sort of a bigger influence than the other, and the direction could change depending on the trait you're talking about or whether it's a male or female offspring.
Yeah,
absolutely. Another thing I've discussed with other people in this general realm of epigenetics and transgenerational inheritance and how it affects cardiometabolic health is actually intergenerational effects. So, for example, there's some really good work out there by a scientist named Bruce Blumberg, who I did a podcast with that I highly recommend to people. And the basic idea there is you expose animals in generation 1 to a chemical, it has an obesogenic effect, so they become fatter basically more easily. And then their offspring see an obesogenic effect, even though they're not exposed to it, and then their offspring and their offspring. So somehow this epigenetic effect can cross generations and persist, even though the subsequent generations were never exposed. And in those studies, it goes out to 4 generations. So, it really is sort of this transgenerational effect. When you look at the experiments you've done, where you look at the effects of exercise, do we see effects that go beyond the next generation and go into the second or the third?
Yeah. So we have looked at the F2. There's a study that is not ours that has looked at the F3, and I believe that the effect is lost by the F3 generation. But what we see with maternal and paternal exercise is that if the F0 mom or dad exercises, you absolutely see the effect conferred to the F1 and then to the F2 generation. But what gets a little bit interesting, and this kind of goes to what we were just talking about, is that so we focus more. My lab is focused more on some of the paternal, to basically grand-paternal effects. So if we're looking at the F0 dads to the the F0 grandparents to the F1 mom or dad to the F2 offspring or grand offspring, when we see an effect depending on if the F1 is a male or female. So the F0 dads to the F1 moms to the F2 male offspring, we see a really striking effect in terms of glucose metabolism. The effect's a little more blunted when we look at the F1 male offspring. So if we go F0 male to F1 male to F2 offspring. And so we're in the process right now of looking at the F1 sperm to see if that has changed kind of in a similar way to the F0 sperm, to see if we can kind of parse apart why some of these things are or are not changing in that realm. From the grand maternal side, Lori Goodyear's lab has done some work with that, and they've seen they definitely see an effect in the F2 offspring, but I believe it's again the F0 grandmoms to the F1 dads to then the F2 offspring, where they're seeing a more striking effect, but they do see it in both.
Mm-hmm.
It's been really interesting to see if what your grandparent did and how that affects your metabolism. And I think as we kind of continue with some of these studies, we'll learn a little bit more about it.
Yeah. And when you study sperm cells, like how do you actually study them, and let's go over like what's inside of sperm cells. These are pretty small cells. They're much smaller than the fertilized egg and the oocytes. What is like the set of stuff in there that could be affected by dad's lifestyle and paternal exercise? Like, are they filled with mitochondria? Do they have a full genome in there? What are the pieces of a sperm cell that are inside of those cells?
Yeah, so I mean, I'll tell you what we've looked at. So, one of the first things we looked at was sperm motility. So we were looking to see, motility changes in response to high-fat diet and exercise, and we do see that exercise increases sperm motility. Again, this is in a mouse model, and high-fat diet reduces it a little bit. And we did try to do a seahorse to measure mitochondrial activity. I'm not sure we were very effective with that. It was kind of our first shot at doing that. But we've not done a ton of things with the sperm other than send it to our colleagues to look at the RNA profiling with it and see what they could see in terms of mapping and getting a library for the small RNA sequencing.
I see. But so at a high level, like motility is affected. Basically, if dad is running more, the sperm are moving.
Sperm are moving faster. I don't think a number was not changed, if I remember correctly. And those were kind of our first two questions, because we were like, how are we affecting conception, or are we affecting conception? Are we changing motility? Are we affecting the number of sperm? And again, in our mouse model, we didn't see any difference in numbers. And then there was a little difference in motility. I don't know enough about that to know if that would be impactful in terms of gestation rates or of conception rates. And we did not see any, again, in our mouse models.
Yeah. What are some of the experiments you guys are working on right now, and the questions that you're going to tackle next?
Yeah. So, with regards to some of these projects, we've gotten really excited at looking at how maternal exercise affects the offspring microbiome. And part of the reason for this is, I mentioned the human milk oligosaccharide component that we saw, 3-sialactose, and it's a probiotic, so it's known to affect the microbiome. What we had seen in some of our animal studies is that when we feed it to mice during their lactation period, so where their microbiome is still forming, it has a really striking effect. What other studies have done, and they've done this in rodents, and in humans, is that if you just give that oligosaccharide to adult humans, it doesn't really make a difference, but their microbiome is already developed. So kind of our question was, is this because we're changing the developing microbiome, or are we, is that increase affecting the microbiome? And so that's kind of what we're looking at now in some of our animal studies, and to see how those changes can be transferred. And we've got some cool data on it. We're seeing some striking changes in the offspring microbiome in both male and female offspring in response to maternal exercise. And then we're doing some fecal microbiome transplants and looking to see how those affect the offspring. So if you take the microbiome from a sedentary mouse and put it in a sedentary mouse, compared to the microbiome from a mouse whose mom was exercised, and put it into a sedentary mouse, what do you see? And so I think we'll learn a lot from some of those studies, and can kind of, work backwards to see how that maternal environment is really, really affecting things.
Yeah. So there's a lot of complexity here, right? Paternal effects, maternal effects, the direction and the magnitude of effects can depend on the trait you're looking at. It can be biased towards the sons or the daughters, depending on which parent you're talking about. It may or may not go 1 or 2 or potentially more generations into the future. So it's a lot of complicated stuff that's going on here. A lot of detail still needs to be worked out. But in general, right, it sounds like when the parents exercise,
it's good.
The things that have been looked at, you tend to see benefits in the offspring.
Absolutely. Yes.
Yeah. Do you have any final thoughts you want to leave people with, or anything you want to reiterate from our discussion just on this general topic of parental exercise and epigenetics?
Yeah, thanks. No, I think this has been a great conversation. I think one of the things that we've been really aware of as we've worked on this is exercise is good. And I never want to say we're going to try to make an exercise mimetic, but I think, looking at ways that we can confer exercise benefits to people who maybe can't exercise is really important. And especially during pregnancy, that's one of the first things that a physician would tell you to stop doing if you were having any trouble, was to not be able to exercise or to reduce activity levels. And so if we can kind of confer some of those benefits to women, I think that would be a really exciting realm for us to take.
Yeah. And so, I would imagine that, a lot of women probably do have trouble exercising just as a natural consequence of the pregnancy. But just to reiterate something you said earlier, I think exercise before the pregnancy can matter. So that's something to be mindful of.
Yeah.
All right. Well, Professor Kristin Stanford, thank you very much for your time. This is fascinating stuff.
Great. Thank you very much.
Hey everyone, a great way to to support my efforts is to visit my support page at mindandmatter.substack.com. A link to that page is in the episode description, or you can search for it on the Substack page itself. The support page has an up-to-date list of my affiliate partners. These are companies I work with, and if you buy those products through those links or using the discount codes provided, those will get you a great deal on a variety of products related to optimizing physical, metabolic, or mental health and vitality. These are all products I use myself, and some of them are directly related to podcast episodes or formulated by Mind and Matter guests. For example, there's a great product called Keto Citra, formulated by a kidney biologist who is on episode number 186. It contains the ketone body BHB together with several minerals in a precise ratio formulated with kidney health in mind. There are also links to products like physical technology devices to track your metabolic health, digital applications to help you find and learn about food and consumer products, and more. One product I use every day is the AquaTrue water filter. I recommend this to everyone because it gets everything out of your drinking water: heavy metals, microbes, endocrine disruptors, microplastics, you name it. Other charcoal or gravity-based filters that a lot of people use don't get everything out, they just get some of the stuff out. So I really like the AquaTrue product. You can look for a link to my affiliate partners on my support page or directly in the episode description to this episode, or you can go to mindandmatter.substack.com to find it there.